Hello, my lovely anatomist and physiologist, Michele Glass here. Thank you for sticking with chapter 10. Our topic here is muscle strength. So what we're looking at with muscle strength is that it's actually related to the number of myofibrils. The myofibril, remember, is the cylindrical shaped structure. It's composed of myofilaments. Myofilaments are your actin and your myosin, your thin and your thick filaments, remember? And the myofibril is organized in sarcomere units. So we can also say it's related to the number of myofibrils and sarcomeres. And what we see is that the number of myofibrils and sarcomeres is affected by hormones. It's affected by stress, and it's affected by activity. And here we're talking about physical activity. So there's that kind of saying, use it or lose it. And that definitely applies in terms of our muscle strength. Use it or lose it. When we talk about changes in the muscle, in the adult muscle, what we're really talking about is changes in the number of those myofibrils. So we have the term hypertrophy. Hypertrophy, the prefix hyper there is like above. So in this case, you're increasing muscle mass. And to increase the muscle mass really means number of myofibrils. So you're not actually changing the number of muscle fibers or cells that you have in an individual muscle. You're changing the properties of the cells themselves, or the properties of the muscle fibers themselves. And so in this case of building strength and building muscle mass, we're really talking about increasing the number of myofibrils. Right, so we're talking about more contractile proteins, more power. The opposite of this is atrophy. And in the case of atrophying, you're decreasing your muscle mass. And again, here, what we're seeing first really is just a decline in the number of myofibrils in individual cells. And so again, this is connected, both hypertrophy and atrophy are connected to hormones, stress, and level of activity. So these are things that can fluctuate and change throughout life. When we talk about the whole muscle, so let's go over here to the side. We had our whole muscle organized into fascicles. And the fascicles, we said, are bundles of muscle cells, what we should be calling fibers, right? So remember, muscle fiber, translate in your mind, that's a muscle cell. So fascicles are at the organ level, the whole muscle level, bundles of those muscle fibers or muscle cells, right? So that's really talking about an anatomical structure. The motor unit, we should think of this more as a physiological structure. Remember where anatomical means what it looks like, what it's called, how it's organized. Physiological is talking about function. So if we call a motor unit a physiological structure, then we're telling something about how the cells actually function, how they work. The motor unit is a group of muscle fibers. You can translate in your mind that that means cells, innervated by the same motor neuron. Group of muscle fibers innervated by the same motor neuron. The term innervated means that the neuron or the nerve is connecting to that tissue. So when we first introduced the idea of muscle contraction, if you remember, I drew this motor neuron. So I drew this long neuron and I had the synaptic knob at the end of the axon. And then I had a skeletal muscle fiber that I illustrated, showing the striations, which is that pattern of the thin and the thick filament that we talked about at the sarcomere, and we saw that it was multinucleated. And we sort of made it look like you had one neuron to one muscle fiber. And that's a simplification. So what we'll see is you have a single neuron, but it has branches to the axon, so that it can synapse on more than one muscle cell. So just like for a little bit of simplicity's sake, I'm going to draw my picture like this, where I have a single motor neuron, but I'm drawing it having 1, 2, 3, 4 different sort of branches. And so I'm showing it like connecting to 4 different muscle fibers. And this is a pretty clumsy picture because it looks like, okay, what's the relationship of these 4 muscle fibers? Why are they all innervated by the same? And so very often you see a diagram that looks more like, instead of looking at kind of the longitudinal view of the muscle fiber, which is what I first drew, you might have a whole fascicle. So I'm going to draw like a big circle to represent my fascicle. And inside my fascicle, I'm going to show some white muscle fibers. And my white muscle fibers are looking just like circles. So I'm seeing the end of the cylinder. And then I'm going to put some pink muscle fibers in the same fascicle here. So I'm doing some pink circles. And then maybe let me do some green muscle fibers. So I have like some green circles in the mix here. So the idea here would be in this fascicle that maybe I have the green motor neuron synapsing it to all the green muscle fibers. Maybe I have the white motor neuron synapsing onto all my white muscle fibers. And then maybe I have my pink motor neuron synapsing onto all those pink muscle fibers. And so the point here is that you have several cells connected physiologically because they're all being controlled by the same motor neuron, meaning if an action potential is traveling on the pink neuron, all of the pink muscle fibers are going to be stimulated. You can have small motor units, and these are going to give you the real fine-tuned movement. So for instance, we can have a lot of dexterity, we say, with our hands or our tongue. We can do very controlled movements of those muscles. So we're going to have smaller motor units there. So that means the ratio of neuron to muscle fiber is a smaller ratio. And so we're getting more fine-tuned control. If we have large motor units, this is going to be larger scale, or we can say gross movement. And remember, in this course, when we say gross, we don't mean like, ew, icky, nasty. We mean big. Say, for instance, postural muscles of my back and neck, they don't need to have like this real detail kind of movement. It just needs to hold my back up, hold my torso up, hold my head up. So I'm going to have large motor units there. Or the powerful muscles of my thigh, gluteus muscles. These are going to have larger motor units than, say, the muscles that are controlling my eyeball, right? And so the motor unit is, again, the number of muscle fibers that are being controlled by the same motor neuron. That takes us to this next bit of information, which is called recruitment. So what we see is that you don't get all those muscle fibers contracting at the same time. So it's not like in our biceps brachii, it's not like we say 1, 2, 3, go, and all of the muscle fibers contract and allow us to, you know, bring our hand up to our shoulder, or they're all relaxing, or like the reverse of that, the triceps brachii is contracting in order to extend the forearm. Is not like that. You guys know this because you're human beings, and so you've had that experience where an object is going to be heavy, and so you know you need to recruit more power in your muscle. Maybe you've felt this experience where you're helping a friend move. They have a really big box. You've been moving a bunch of heavy boxes all day. You see that big box and you're like, I'm ready, I'm gonna do this. And so you bend at the leg and you're like recruiting your legs, and you go to grab that box, and then like you fall over because instead of being full of books, it's full of pillows. And so it's really lightweight. You over recruited. So your brain said, oh, this is a big box. All these boxes have been heavy. This will be heavy. Recruit a whole lot of muscles, right? Or maybe the flip of that, where you're like, oh, let me help you carry your bag. And so you go to pick up somebody's backpack thinking that it's just got like a notebook and a pencil in it. And then you realize, oh, crud, the whole anatomy and physiology textbook and the microbiology textbook are both in there. And that sucker is heavy. And so you have to actually kind of like set it down and re-recruit because you under-recruited, right? And so just our experiences of life teach us like how much muscle we should recruit so that we don't have to consciously do a whole processing typically. So recruitment is talking about the number of muscle fibers, or we could even say motor units contracting. And as you increase recruitment, meaning you get more muscle fibers, more motor units online, you're going to increase the amount of tension or the power that you can produce in that muscle. So there's a direct relationship here. Both arrows are going up. The more muscle fibers recruited, the more power or tension you can build in that particular muscle. What we see too with recruitment is we have what's called asynchronous recruitment. And so in this case, we see that some motor units are at rest while others contract. So instead of saying, okay, 1, 2, 3, every single muscle fiber contract, and then they all fatigue and you have to wait, you let, you know, group A contract, and as they're relaxing, you let group B contract, and as they're relaxing, you let group C contract, and then they can relax. And so you're able to sort of maintain tension or power in that muscle longer because you're alternating which motor units are being activated. The last thing to talk about in this particular presentation is the length tension relationship. Okay, so what we know based on our sarcomere unit, you have to have overlap between the actin and the myosin, or you could say between the thin filament and the thick filament, in order to get cross bridge formation. Cross bridge formation, remember, is talking about when that myosin head reaches up and grabs hold of the actin. That was the formation. So clearly, in order for a myosin head to grab hold of an actin, there has to be one in proximity, and that's what we saw in that zone of overlap. Now, there's an ideal amount of overlap, and that's like 80% to about 120% of the resting length. So if the sarcomere unit is at this ideal length, then we're able to produce a good amount of tension. If there's no overlap between the actin and the myosin, they're not going to be able to do any cross bridge formation. If there's only a tiny bit of overlap, then you have to do a whole lot more contraction to get to the ideal before you can build tension. And if you've already like completely slid the actin all the way in towards the M line, there's no more room, there's nowhere else to go. So if you have too much overlap, you're pretty much done contracting. If you don't have enough overlap, you got to work a little bit to get to that ideal point. So if it's stretched, if it's overstretched, if it's stretched past that ideal, that means you don't have enough overlap. In order to get to that cross bridge formation. Now, usually this doesn't happen. This is prevented by the tendons and ligaments connecting. Remember, ligaments are connecting bone to bone. The tendon is connecting the muscle to the bone. So these structures are important in helping to stabilize our joints and our muscles. And we see that because of these relationships, we don't typically get this overstretch. If it is shortened beyond that 80%, I put is all jammed up. Meaning like those actin are bumping up against the M line. There's just not anywhere else to slide that thin filament across. So you really start to have a whole lot of problem. You have nowhere for them to slide. So what we see with the length tension relationship is if you're at your ideal length, you're going to get your like maximum tension. If you're overstretched or understretched, too much or too little overlap, you're going to see problems in establishing the tension. Okay, the other factor that affects tension in a muscle is frequency. So stay tuned for the next video. As always, take care of yourselves and each other.